Hi everyone, my name is Connor Fox and I am a senior at Radford University studying biology. This summer I had the opportunity to do the SURF with Dr. Justin Anderson, and my project is Inducing Mutagenesis in Phage HanSolo with hydroxylamine. So before I get into the main body of the project, I did want to go over some background information that explains why this research is relevant. So Dr. Robert Redfield, who's the current director of the CDC, has described the time we are living in as a post-antibiotic era. And what he means by this is because of overuse of antibiotics, they're becoming less useful to treat bacterial infections. They've had the chance to evolve and become resistant to many of the drugs we use. And currently there are 18 antibiotic-resistant pathogens that are considered threats to public health. And all of this culminates in 2.8 million antibiotic-resistant infections annually in the United States, and this unfortunately results in 35,000 deaths per year, which that's a lot, and that's very scary. I've put a few common antibiotic-resistant bugs in the image on the side. There's the Acinetobacter, there's MRSA, there's Clostridium difficile, and drug-resistant gonorrhea. So because antibiotics are becoming less useful, it's important to explore new ways to treat bacterial infections. And one of these ways is bacteriophage therapy. And I will get into what a bacteriophage is in the next slide, but before I do, I just wanted to list two examples of successful use of bacteriophage therapy in medicine. In the top image, you can see Dr. Graham Hatfull in the left middle, sitting with the patient he helped to treat. He is a biotechnologist at the University of Pittsburgh, and he was contacted by a doctor in London who had a cystic fibrosis patient that had just undergone a lung transplant. As a result of that lung transplant, she contracted a Mycobacterium abscessus infection that was not responding to any antibiotics. So the doctor reached out to Dr. Hatfull, who is pretty notable in the phage biology community. He has a massive archive of phages. And he asked him, do you have any phages that could be used to treat her infection? He did. He had 3, so he helped create a phage therapy cocktail that successfully treated her infection. The below picture is Dr. Stephanie Strathdee of the University of California, San Diego. She is an epidemiologist, and when her and her husband were on vacation in Egypt, he contracted an Acinetobacter baumannii infection, which also was not responding to any antibiotics. She was desperate to save his life as he was not responding to any treatment, so she came across phage therapy and she begged doctors to try it. And she led a team of doctors and scientists to save her husband's life using phage therapy, which was pretty amazing. So what is a bacteriophage? Put simply, it's just a bacterial virus. Phage comes from the Greek phagein, which means to eat. So you can also call it a bacteria eater virus. The top image is an electron micrograph of a phage. You can see a protein capsid which contains the phage's genome, and then a tail which has some proteins on it that help it attach to a host. So for the relevance and sake of this presentation, we are only going to be going over the lytic life cycle, which I put a little graphic of in the bottom. So during the lytic life cycle, a phage particle will attach to a host and insert its DNA through the tail into the host cell. This DNA then gets incorporated into the host genome, and when the host reads it, its biomachinery starts to make the necessary proteins like capsid and tail fibers and DNA necessary for individual phage particles, and it begins to assemble them. Once enough phage particles have been assembled, they will kind of explode out of the cell in a process called lysis and jump to new host cells. And it just continues to repeat this process. So the advantages of using bacteriophages in medicine is that they are natural and they're not antibiotics, so you don't have to worry about antibiotic resistance. If you've ever been on an antibiotic and have experienced unpleasant side effects, this refers to toxicity. Bacteriophages don't have that same unpleasantness associated with them. They're low cost, and another really important thing is that because they have such a narrow host range, they're only going to interact and kill the pathogenic bacteria affecting you. They're not going to disrupt your normal microflora, which is extremely important. And then another nice thing is that they're pretty easy to rapidly discover. A few potential disadvantages: not all phages make good therapeutics. The narrow host range can be problematic depending on the species of bacteria, but the primary problem is the unfamiliarity with phage biology. So we need to do a lot more research before we can implement bacteriophages into medicine. They were only discovered in 1915, so we only have known about them for 105 years, so we need to do more research. So my project is inducing mutagenesis in phage HanSolo with hydroxylamine. So we are going to introduce the mutagen hydroxylamine to the phage, and we are going to then plate these mutants and collect them. We're then going to extract their DNA and compare it to the original DNA to see if there have been changes. So the purpose of doing this is to better understand what genes contribute to a phage's overall viability and infectiousness. And how I explain this is, if you think about a human being, we know what certain organs do in our body. We know that the brain controls thought and motor function. So we know if someone has a seizure, or if someone is slurring their words after a head injury, we know that something is not working in the brain. We know that something has happened, and that's causing that effect. By mutagenizing this bacteriophage HanSolo, we are going to observe the effects it has on the phage's infectiousness and its ability to reproduce. And then when we extract that DNA, we are going to compare it to the original host genome. And we are going to look for any differences. And if there's a mutation in a certain area, we're going to be able to say this mutation in this gene causes this effect on this bacteriophage, which is going to kind of tell us what genes are really important to how well a phage can infect and survive. So the bacteria we are working with is HanSolo, that's the name. Its host is Microbacterium foliorum, and it was isolated by myself and my lab partner Thomas Marshall in Radford, Virginia in 2018. Its morphotype, which just refers to the shape of the viron, is Cephaloviridea, and its plaque morphology, this just refers to the image in the top, where you can see a plate with a bacterial lawn and a bunch of circular clearings. That's a plaque, and they're just small and circular. So our hypothesis is that when exposed to hydroxylamine, HanSolo will undergo mutagenesis, which will lead to changes in plaque morphology, indicating altered growth patterns. Plaques are very specific to certain phages, and they're kind of a primary indicator if you have multiple phages in one sample. So if we see changes in the way HanSolo's plaques look before, like after it's been exposed to the mutagen, that indicates that a mutation has occurred, causing a change in how it reproduces. So here's a little graphic of the basic methodology. So we're using basic molecular biology techniques to amplify the phage. We already had a kind of like a pretty high titer stock of the virus from 2018. So I just amplified that using serial dilutions and collecting the product on a plate using a buffer. And then after we had a high enough volume, I took a portion of it and exposed it to the mutagen, hydroxylamine, and then plated this virus and kind of observed if there were any like noticeable differences between the original HanSolo and the post-mutagen HanSolo, and there were. So I collected these mutants and I amplified them in the same way I did with the original, and I am currently working on isolating their DNA, and then once I have completely successfully done that, we are going to sequence that DNA and compare it to the original DNA of HanSolo. So, plaque morphology is a pretty important topic in phage biology. A plaque, like I said before, is just a clearing of cells on a bacterial lawn caused by viral growth. And plaques are influenced by the type of phage, the host, and various growth conditions like nutrients and temperature and time. And different looking plaques, like I said, indicate different phages. So size, turbidity, shape are all things we look at when observing plaque morphology. So, like I said, so far I have successfully isolated 3 potential mutants, and I am currently working on extracting DNA from them, and then we will move on to annotating their genomes. But I have plated them, and as you can see, sorry, I'm moving the webcam so you can see the picture. There is a noticeable, discernible difference between the wild-type HanSolo and the mutants. You can see, looking at the plaques in proposed mutant 1, the plaques are bigger. In proposed mutant 2, they have halos around them and they're a lot more turbid, which just means they aren't as clear. And then in proposed mutant 3, it's kind of a mix of both. The image on the end is of what's called a spot test, where I just took 3 microliters of each of the lysate and just spotted them on a plate with a bacterial lawn and just watched how it grew. And you can see for the wild type, it's pretty circular and even with nice clean edges, but all of the mutants kind of have jagged edges and don't look as clean. Some further directions I would like to go in is observing some plaque morphology, just in how it changes over time. I noticed during the course of the project that plaques change drastically as they're developing, so that'd be another area I'd like to look into. I also am really excited to work on annotating the genomes and comparing that with the wild type and seeing if there are any major changes and where they are. But I appreciate the opportunity to present my research to you all, and I welcome any questions during the live Q&A. I want to say thank you to the Office of Undergraduate Research and Scholarship, particularly Dr. Virgo and Dr. Pate, for their continued support and guidance during this project, the Radford University Department of Biology, Dr. Justin Anderson, who is my mentor, and the SURF class of 2020. Congratulations everyone on your research, and thank you all for watching.